Okay, to begin with, I want to just to thank all the members who have worked really quite hard to get all this together. We have quite a few members in the project. Texas A&M, of course, Plant and Food Research out of New Zealand, Cornell, Wisconsin, Pennsylvania, Wageningen from Netherlands, Washington, Minnesota, North Carolina, Arkansas, Maine, and Oregon. So it's really quite a national and international affair here. And I also want to thank all the other collaborators, all the commercial firms that have helped us and supported us when we're getting this grant funded. Remember, when we do these, we have to get all sorts of letters of support to convince the reviewers that it's a worthwhile project, and we did very well in doing that. Also, like to particularly thank various firms because they contributed to our fund to offer travel scholarships, and that'd be Star Roses and Plants, Gene Twister, Bayjill, Thumb and Orange, Newhouse Farms, and Arbor Biosciences, and Pairwise. And I want to thank Illumina because they donated some materials for sequencing for our lab too. But all the other people have been very supportive in trying to get this up and going, and it's been a lot of fun to work with everybody. Of course, I'm David Byrne. I'm the coordinator here, and nice to actually see people in person finally. It's been a long time coming. And those online, hopefully you can hear me. If you can't, somehow tell me. And we will try to figure out those technical glitches if they're there. But I'm at Texas A&M. You can go to tools for polyploids at tamu.edu if you want to email me, and I'll get those emails. And the other thing we want to discuss at this meeting, since we're finally together and we're about halfway, a little over halfway through the project, is what the next step for Tools for Polyploid projects will be. And we'll have these discussions informally right now as we go through the day. We have Oscar and Jeff, our co-PDs here, and they will probably linger around. There's tables out there on that side where you can get around and discuss things during breaks and lunch or whatever. And so I encourage you to do that. Tonight, there is sort of a planning dinner that we'll, again, socially meet and discuss these things as well, sort of get a beginning look at what we want to do as we go forward. Because this is the third meeting, we have one more training workshop to go, and then the project more or less finishes. So, but I think we have a lot more to do, so we'll have to see what we want to do in the future. Now, as you know, this project has three different major objectives. One is just developing computational tools, and every year we've been learning about new computational tools that have been developed. And then the other part is actually validate and test them. So we have a group of breeders that do that. There's a lot of interaction between the breeders and the computational scientists, programmers that sort of get things working well for the breeder and breeding program per se. And of course, the last objective is what we're doing now is the training. And so that all those are very important. You want that, huh? Okay. Yeah, of course, again, the next step, what we have to do is figure out the next steps in tools for polyploids. And so encouraging, we have discussions during the day, objectives, what crops, what workshop objectives, things like that, formats, anything you want to change, we can change. Okay, so what I want to do now is just quickly go over who the polyploid crop community is, like I do every year. And so when we do registration, I always ask you for all these details, which is, you may think is not important, but for me, as reporting what we're doing, this is extremely important. So when we tell people that we're doing this and who's involved, usually you don't get this type of information in these projects. So this is very important as we go forward. Again, age-wise, as before, 60, 70% are younger than 40. I'm one of the oldest ones here, and I'm glad to see there's young people here learning. because we got a long ways to go as we go through here. But it's been pretty steady throughout the three workshops. Who's participating with respect to private versus public versus students? So we have about 20 or so % private breeders participating in this meeting. 35 to 40% are public breeders, and then the rest are students and postdocs, which is what I sort of expected with respect to students and postdocs being very heavy there. I would like to see more private participation in the training workshops too, and we'll have to talk about how to get that done as well. So we're people from about half from North America, which makes it extremely international from a specialty crop research initiative proposal, because normally it's mainly North America. So it's about half from North America, and then of course South America. Europe is fairly heavy. Africa, quite a bit. Asia and Oceania, New Zealand, Australia, there's a few there. And of course we have some visitors with us as well. One thing that we're going to be doing in conjunction with the Rosacea Genomics Conference, which is going to be in Nelson, New Zealand in March, is to have a one-day workshop on tools and polyploids. I figure that's sort of a completely, it's kind of hard for them to come to these, virtually and even physically. So we thought we'd go ahead and see if there's much interest. And since we're going to be there anyways, we thought we'd put this workshop on and see how that works. And it'll be available both in person and online. There'll be more information as we go on. If you look at the expertise represented, now I make these big groups, but you get a general idea. A lot of breeders, computational geneticists and molecular geneticists are about equal. And now this is, when I ask this question, people chose various categories. So this is not a one-for-one, it's expertise is out there among all this group. So, and then plant pathology, entomology, and plant physiology, we have quite a bit of expertise in there too. So it's really a wide, broadly educated group here. As before, root and tuber crops and fruit are the most represented. Of course, root and tuber crops, you're talking about potatoes, sweet potatoes, and yams mainly. Fruits, you're talking about mainly the berries. Grass, a lot of turf grass, but this also includes sugarcane. I put them in there and a few other things. Forage grasses would be in there too. So although it started as a horticultural, especially crop thing, we're getting other people joining in as they can, because they realize it's pertinent to more than just horticultural crops. And of course, vegetables and cereals are represented among the people attending as well. Ploidy, again, mainly tetraploid, but realize that most people working in polyploid crops work with multiple ploidy levels. Many do work with diploids as well. So that's where you get the diploids in there. But also the software works well for highly heterozygous, diploids as well, especially the clonally propagated. So that's where we are ploidy-wise. How about the use of the software? This is, again, a question I ask every year, because I'm trying to see if there's any trends. Hopefully it's going up as we go along. And if you look at it, it is, which is, it's a good sign. So we're doing some good somehow. And so as you look across it, it's generally the trend is you're getting more used. And if you look on the right-hand side with reads to map and over, those are ones that we're just introducing, so we don't have all 3 years of data on that. If you look at a little more closely, just the ones that we've looked at from the beginning, you can see in 21, 11% of the people that assisted were familiar with it. By 22, we're up to 21%, and now we're up to an average of 29% familiar with the software. So again, year by year, we're getting more and more activity here. And if you just look at the software that has to do with mapping and QTL discovery, you can see the averages are a little different. 11% in 21, in 22, we're up to 24%, in 23, we're up to 36% of our group are familiar with these types of software. So again, improvement is what we're hoping to get, and hopefully that will go even higher as time goes on. Okay, so the other question is, are people actually using this stuff for genomics-assisted breeding? So that's another question I always ask. And so is it increasing? Yeah, it does seem to be increasing. If you look at this, this is molecular marker used for parental selection. And you have never, which is the percent of people that never have used it is going down. And the people that are always using it, about the same, but you're getting up in the sometimes in the 50% of the time. So again, we're getting a trend to use it more often for parental selection. You see the same trend for seedling selection. And of course, if you go to genomic selection, the same sort of trend, though much less, many fewer people are using genomic selection. Okay, so I want to thank the people who did the December sessions on computational sessions. And we had 3 sessions that were extremely successful. Maria, Gabrielle, Jekin, and Chris were wonderful. We had, what we did this time, we did an overview of these topics first and had some examples. And through those examples, we helped people go through the examples, answer questions as we went along. And it worked out really quite well. We had about 50 participants every time. So I think it was substantially useful. So just quickly with respect to the program today, the first day, we're going to be, the major training presentations are on reads the map, 2 approaches for QTL analysis with interconnected or multi-parental families, and then marker imputation in polyploids. The second day, we're going to look at software comparisons, QTL analysis, genomic selection, interactions, polyhaplotype haplotype assembly. You have genomic selection and breeding strategies for clonal crops. And this is interactive. I've seen this presentation before. It's really quite fascinating. So I think you'll all enjoy this, or a similar one, if not that one. And then we also have, as we did before, oral presentations that are interspersed between the major presentations to give you an idea what other people are doing and what results they're getting. And then we'll have poster presentations as well. And the poster session is this afternoon. So this is the agenda today, with the specific talks. I won't go through a lot of details here, just so you know what's coming up. These are the user presentations. So we have 5 user presentations that will be oral and be interspersed. And then we have our poster presentations. I just found out that one of the poster presentations, Mr. O won't be here because he had some airline issues. I gather a lot of people did, but I'm glad this many made it in spite of the challenges. So he's going to have a digital presentation. Not quite sure how we're going to do that, but we'll figure it out. The second day, again, we went through this, and these are specific presentations we have on the major presentations. And these are the user presentations. Again, we have 5 that we have, again, on a wide range of topics. So it should be very interesting to hear their perspectives and their progress. And then the poster presentations, we have 6 here that will be available.